Nucleotide synthesis. Nucleotide synthesis involves two syntheses, that is, purine and pyrimidine synthesis. In order to make them, we need to look at pentose synthesis first. Pentose synthesis. Once glucose enters the cell, there's a different types of enzyme. For example, hexokinase enzyme. Enzyme hexokinase phosphorylate glucose at carbon number 6. ATP was added to give 1-phosphate and convert into ADP. This glucose was converted into glucose 6-phosphate. Glucose 6-phosphate can undergo either glycolysis pathway or pentose phosphate pathway. If this molecule enters pentose phosphate pathway, there's enzyme called glucose 6-phosphate dehydrogenase. An ADP positive was added to take hydrogen and convert into NADPH. Then, glucose 6-phosphate was converted into 6-phosphoglucolactone. We add some water to this reaction and enzyme lactonase involved in this process. Then, we convert 6-phosphoglucanolactone into 6-phosphogluconate. In this step, enzyme 6-phosphogluconate dehydrogenase involved. NADP positive convert into NADPH and take 1 hydrogen. 6-phosphogluconate was converted into ribulose 5-phosphate. Here, we should know that ribulose 5-phosphate is a ketone. We need to convert ketone into aldehyde, so we use enzyme isomerase. Enzyme isomerase convert ribulose 5-phosphate into ribose 5-phosphate. Ribose 5-phosphate is a aldehyde. Then, ribose 5-phosphate, we need to transfer this molecule into ring. So we use enzyme ribose phosphate pyrophosphokinase. Then this enzyme convert ribose 5-phosphate into the ring and called phosphoribosyl pyrophosphate, or abbreviated as PRPP. We also add phosphate group using ATP. So here ATP was utilized into AMP. Then this PRPP can convert into two different sugar. The difference between two sugar is hydroxy group and hydrogen group. If their hydroxyl group on second carbon, then this sugar called ribose sugar. When hydrogen group was at second carbon, so it called deoxyribose sugar. So we started pyrimidine synthesis. The first step of pyrimidine synthesis involved 3 molecules, that is glutamine, bicarbonate, and 2 ATP. We use 3 of these molecules because we need from glutamine, we need amino group from glutamine, we need double bond of oxygen from bicarbonate, and we need to add phosphate, so we use ATP. Then we add some water and use enzyme called carbamoyl phosphate synthetase type 2 for this reaction and convert these 3 molecules into one molecule called carbamoyl phosphate. Then, carbamoyl phosphate, we must add carbamoyl phosphate with aspartate. To add aspartate, we need to use enzyme called aspartate transcarbamylase. Then, carbamoyl phosphate add aspartate and convert into carbamyl aspartate. So here, we need to dehydrate this molecule, so we use enzyme dehydratase. Enzyme dehydratase pull out water from this molecule, and this molecule was converted into orotate. Then, in this process, we must add PRPP. That PRPP we show at pentose phosphate pathway. To add PRPP, we need some enzyme called orotate phosphoribosyl transferase. Then, orotate was added with PRPP and converted into ornithidine monophosphate. So here, we need to chop off this carboxyl group. So we release in the form of CO2 using UMP synthase. This enzyme decarboxylated ornithidine monophosphate and convert into uretidine monophosphate. Then, enzyme nucleotide diphosphate kinase convert this UMP into uretidine triphosphate. So, uretidine triphosphate has 3 phosphate group here, and we need to convert uretidine triphosphate to cytidine triphosphate using glutamine. Here at this process, cytidine triphosphate, we need to convert into cytidine monophosphate. So we need to pull out this phosphate group. So we pull out pyrophosphate, and this CTP can convert into CMP. Then CMP or cytidine monophosphate can be used to convert into RNA. Same as here, same as UMP, UMP also can convert into RNA. So here, this cytidine monophosphate can convert into RNA. So how do we can convert this CMP into DNA? We need to use enzyme ribonucleotide reductase. This enzyme can add phosphate group here and convert this cytidine monophosphate CMP into deoxycytidine diphosphate. Then this DCDP was pull out one phosphate group and convert into deoxycytidine monophosphate or DCMP. The phosphate group was pulled out, and this DCMP can convert into DNA. So another way that can be used here is we started at uretidine monophosphate UMP. In this process, we use enzyme ribonucleotide reductase. This enzyme add phosphate group here and convert this UMP into deoxyuretidine diphosphate or DUDP. Then DUDP was pulled out one phosphate group and convert into deoxyuretidine monophosphate or DUMP. And the next process, we need to add methyl group. So we add tetrahydrofolate. If we want to add methyl group here, we need to use enzyme thymidylate synthase. So this DUMP was added with tetrahydrofolate and convert into deoxythymidine monophosphate or DTMP. So this molecule can convert into DNA. We move to the next synthesis, that is purine synthesis. Here at purine synthesis, we use some molecule that is PRPP, phosphoribosyl pyrophosphate, glutamine, bicarbonate, formate molecule, glycine, and aspartate. So we convert this molecule into one molecule called inosine monophosphate or IMP. So this synthesis has 10 steps. So this molecule called IMP, the first step, we use inosine monophosphate dehydrogenase enzyme, and NAD positive was converted into NADH. So this IMP was converted into molecule called hypoxanthine monophosphate. Then hypoxanthine monophosphate was converted into guanosine monophosphate using GMP synthase, and glutamine involved here, and glutamine was converted into glutamate. So this guanosine monophosphate or GMP can be used to convert into RNA. Then we need to convert into DNA too. So we must add phosphate group here at GMP. So we use ribonucleotide reductase. Then this GMP was converted into molecule called deoxyguanosine monophosphate DGDP. So this DGDP, we need to put out some phosphate group. So this DGDP was converted into deoxyguanosine monophosphate DGMP. So this DGMP can be used to convert into DNA. Then here, in this IMP molecule, we need to convert into adenylosuccinate. So to make this molecule convert into adenylosuccinate, we need to add aspartate. So when we add aspartate, we need to use enzyme called adenylosuccinate synthase. Then this IMP was converted into adenylosuccinate. Then we need to convert adenylosuccinate into adenosine monophosphate. So we need to cut off these things. If we need to cut off this part, we cut off in the form of fumarate. When we cut off this part, we use enzyme adenylosuccinate lyase and convert this adenylosuccinate into adenosine monophosphate. This adenosine monophosphate can be used to convert into RNA. This adenosine monophosphate was converted into deoxyadenosine diphosphate using ribonucleotide reductase. So here, this enzyme add some phosphate group here to convert this AMP into DADP. Then this molecule was pull out one phosphate group and convert into deoxyadenosine monophosphate, or called DAMP. So this DAMP molecule can be used to make DNA. That's all from me. Thank you.